Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I'm very happy to bring the conversation I had with Michel Brahic. Michel is an expert in viral and prion infections of the brain. He's also an honorary professor at Institut Pasteur in Paris. He's also been a consulting professor at Stanford University School of Medicine, and his work has been published in many journals such as Nature, Cell, New Scientist, and others. He is the author of the latest book, The Power of Prions, the strange and essential proteins that can cause Alzheimer's, Parkinson's, and other diseases. We talk about prions and many of the other components that are implicated in where we see prions most salient in certain diseases. We start by giving an overview of proteins. We talk about prion proteins. We talk about Creutzfeldt-Jakob Disease and the importance of cells and ribosomes. We also talk about some parts of the brain, some of the neuroanatomical regions relevant for prions. Talk about Lewy Body, Alzheimer's, Parkinson's, memory, the future of medical research on prions, and many other topics. This was a fun one. I've done in my own clinical work some experience and training in neuropsychology, so this was fun to kind of scratch that itch a little bit and talk about the brain and talk about proteins and talk about many aspects of many commonly known diseases, which are awful for those that have them. And it is really the important work that he has done and many others to try and help us find cures and or treatments for things like Alzheimer's and Parkinson's, which are quite debilitating. As always, you can find this conversation and all other conversations at convergingdialogues.substack.com. Also on YouTube, like, subscribe, share with your friends, all the wonderful things. And now I bring you Michel Brahic. I'm here with Michel Brahic. Michel, thanks so much for coming on the podcast. I'm looking forward to speaking with you.
Thank you very much. Happy to be here.
Yes. You have a mighty little book, little in terms of the size. Princeton did such a nice job with the how they put this together. It's a kind of almost like a coffee table size book, but has so much wonderful information inside of it, so many good things to learn. The book is called The Power of Prions, the strange and essential proteins that can cause Alzheimer's, Parkinson's, and other diseases. And again, this is out through Princeton. It's very good. So we'll talk all about prions and many of these neurodegenerative autoimmune diseases. But before we do, just tell listeners just a brief snapshot of who you are professionally, academically, and all the relevant details there.
Okay. Well, actually, I am an MD, PhD, in fact. And my career has been entirely focused on the brain, or the central nervous system, and its infection by viruses primarily. But then I've always had a very strong interest in the field of prion, even before they were called prion, when I was a medical student. And more recently, about 10 years ago, I got specifically involved in a prion protein called alpha-synuclein, which is involved in Parkinson's disease. So, that's about what I am. So in terms of profession, I've had a career which has been going back and forth between the US and France. I was at UCSF in San Francisco, University of California, San Francisco, for several years. That was actually my first faculty appointment. Then I moved back to France, and I spent more than 20 years at Institut Pasteur in Paris, where I kept going in the same field that I just described, until I had to retire because I had a mandatory retirement at 65. I had organized something in advance to be able to join Stanford University, and I arrived there as a consulting professor to follow my research. I had some American grants that I could take with me and continue working on them, on the project. And it's during my time at Stanford that just by chance, and discussing with a colleague and a friend, that I realized that what we were doing at the time, using all sorts of techniques to follow what viruses do to the brain, could be applied now to the field of prions, because it had just been suggested that prion protein could be also responsible for neurodegenerative disease, in particular Parkinson. And so I established a collaboration with other French scientists who happened to be on sabbatical at Stanford at the time, Ronald Melky, to essentially use the technology that we were using for viruses for this protein alpha-synuclein, following what happens when you inject it into the brain of mice, and also when you add that protein in its prion form to neurons that we grow in culture, and so follow the entry, the progression of the diffusion of that protein, both in vivo in the animal and in vivo, but in tissue culture, in neuron culture. So that's, in a nutshell, what brought me to write this book, because I was teaching a class at Stanford until 2 years ago on the subject, and I thought it would be a good idea to write something not for my students any longer, but for a lay audience, about an aspect of biology which is not very well discussed or appreciated in a lay audience, maybe. So I thought they would feel the need, hopefully also excite some curiosity in young scientists who weren't aware of this field, or young people who want to become scientists. Yeah, that's about my story.
Yeah, I think that's spectacular. I mean, I think it is. These things are very important, and we need more people to understand kind of the workings of some of the things that can arise for people when they get these illnesses, unfortunately. So proteins are super essential. That's the big thing of the book. So before we can talk about any of this stuff, maybe just kind of give us the kind of brief snapshot of proteins, what they are, and how they're so important for so many aspects of life.
Well, life, there would be no life without protein. I mean, proteins are one of the essential components of life, all the way from viruses to us. They serve an incredible number of different functions. And they are the product of our genes. So the genes are, in fact, contain a code which tells the cell how to translate this code into a protein. The protein itself is a big molecule, big by comparison with some general molecule, but it's not big enough to be seen, for example, in an electron microscope. But they are made of a string of smaller molecules called amino acids, which are linked to each other in a chain, which can be more or less long, sometimes very long, some are shorter. And that chain, there are 20 different of those small molecule which makes the chains. So between the fact that the number of subunits of small molecules is 20, the fact that the order in which they are in the chain depends changes from one protein to another one. It's specific to a particular protein. And also the fact that that chain, which is linear, will have to fold into a three-dimensional structure to get its shape. So because of all these variables, the order of the amino acid, which is called the sequence, the length, and then the folding, you have an enormous variety of structure at the end. And this is why proteins are performing so many functions, because of this fabulous possibility of having different shapes with different chemical property at their surface or inside. And they can do all sorts of different jobs. And they do perform an enormous number of jobs in our cell or in life in general, even viruses which are not cells. So that's what proteins are. To really appreciate what a protein is, you really need some time of a drawing or a picture, and there is one in this book, to appreciate the three-dimensional aspect of a protein.
Yes. Some of the diagrams and graphs in the book are very helpful in trying to understand some of these things. And so, obviously, the proteins become important with the folding. And so, as you just nicely explained there about proteins, one of the things that we understand, like we do with many diseases, is there's something that goes wrong, or there's something that doesn't completely connect, or something that's defective in some way at the protein level. So you talk about prion diseases. Now, this is interesting. Maybe if you can kind of spell out for us the kind of taxonomy of prion diseases, right? You start in the book by talking about scrapie, and then Kuru, and then the other disease that's well known. It's got an interesting name there. And we also talk about mad cow disease. All of these are kind of created by PRP, which these are prion proteins. What's the taxonomy though? Like when we're thinking about prions diseases, how do we understand which goes where and how the kind of structure or hierarchy is here?
Okay, that's a very important question. First of all, let me say in advance, and we may come back to that later in the talk, that prion proteins were discovered because they are the cause of some diseases. But they are also essential for life. They perform some functions which we are now discovering. So it's a very large group of protein. Now, to talk about taxonomy is probably not exactly the right term, because it suggests affiliation between the different proteins. In fact, many of those proteins are genetically unrelated. They are different proteins in the genes without taxonomic affiliation. So it's a question of more of definition. And that's very important and can be very confusing. The term prion was coined by Stanley Prusner to describe the agent of strange diseases, Cru in New Guinea, Creutzfeldt-Jakob, which is worldwide, and scrapie of sheep, and some others. So all of those diseases, which altogether are called spongiform encephalopathies because they create tiny holes in the brain of the patients, which eventually can look like a sponge a little bit. All those diseases are caused by the same protein, PRP, which can acquire a prion activity. PRP is a normal protein of our cells. Its function is not entirely understood, but it can take a different shape from its normal shape, and then it becomes the cause of those diseases. Now, the other prion protein that we already mentioned, the one alpha-synuclein for Parkinson, A-beta and tau for Alzheimer, and some others, they are totally unrelated to PRP. They are very different in terms of structure, etc. And we can call them prion because they behave in their folding in the same way as PRP. But it could be considered as not very clearly use of the term prion, and some people prefer to call them prion-like, for example, because they behave like prion, but they are not PRP protein. They are different protein from PRP, alpha-synuclein, A-beta-42, etc. So it's really a question of definition. What do we want to call prion? In the book and in conversation, we tend to say prion for everybody because of all those common properties they have. But originally, the name prion was given specifically to the PRP protein, the one responsible for Creutzfeldt-Jakob and scrapie in sheep and mad cow disease and some others.
That's a very, very important distinction there that you're making between, because I was a little kind of unsure about this myself well, we have PRP, prion proteins, but how are they different or linked or not linked to other neurodegenerative diseases such as Parkinson's, Alzheimer's? And so you're explaining there how it's like that, but it's not quite that, which is very helpful. You did mention, though, Creutzfeldt-Jakob disease, which I'm curious about. This is it's super rare, and this also seems to have similarities with some, scrapie and other things, but it seems to be somewhat different. I guess, how do we see, if we do, the prions implicated in this rare disease?
Okay. Yeah. So let me make absolutely sure. I mean, PRP is responsible for those spongiform encephalopathy, including Creutzfeldt-Jakob disease, mad cow disease, etc. The other ones, Parkinson, etc., they are totally different proteins. What they have in common, and the reason, I'll just go back a little bit to that, and I will come back to Creutzfeldt-Jakob after that. What they have in common, and the reason for which we call them prion, all of them, or prion-like for the other one, is that the fact that they have become the agent of diseases, or for them, the fact that they perform a very important function, is because they have different way of folding. And in the case of the one which are called responsible of diseases, like PRP, we talk about misfolding, which means that they don't take their normal folding, the one which gives them a function for the cell, but by accident, and it's a little bit complicated to explain like this again without making diagrams or having pictures, but by a sort of accident, which is really due to chance or malchance, in fact, they can acquire a different folding, which makes them responsible for a disease. So Creutzfeldt-Jakob is therefore one of the ones which are caused by PRP, the original prion protein. It's a rare disease. It's only one case per million and per year. It's worldwide. And it causes dementia, which is a loss of, as we know, of higher function of the brain, including memory, etc., etc. It's always fatal, unfortunately. There is no treatment. We haven't found any way to prevent these diseases. So neurologists know about it, but neurologists in practice don't see many of those because it's one case per million and per year. But there are centers which are specialized in dealing with those diseases all over the developed world. What else can I say about Creutzfeldt-Jakob disease? There are some forms, so it's a very rare disease, and there are some forms which are familial, so they are genetic. They are not sporadic. The sporadic form, the one case per million per year, again, are due to chance misfolding of the protein. The genetic form are due to mutation in the gene of the protein, which favors this misfolding, so that the protein is almost invariably going to misfold at some point during time in the brain of the patient who has the mutation. and then cause the disease. So that's about 10% of the Creutzfeldt-Jakob disease, which are genetic. So it's 10% of 1 million per year, so it's extremely rare. But some families have been identified all over the world, and their genes been sequenced, and all those cases which are familiar have mutation in the gene that code for the PRP protein.
It's very interesting how, as folks like yourself are doing research and you're understanding like how proteins can look and fold and manifest in different ways, how they can be able to contribute in some ways, but then also kind of work against us as well in other ways, which is really, really interesting. Before I guess we move on to some of the things in Parkinson's, maybe just one thing here, if you want. We talked earlier about amino acids and proteins, and even in the book, you talk about alpha helices and beta strands and loops. You can talk about some of that if you want. But I guess the one thing I'm curious here as it pertains to some of these diseases, how do we see the centrality of cells and even ribosomes and how they play in making proteins, and why this is important to kind of recall here when trying to understand some of these diseases?
Okay, well, this is I mean, in order to understand the disease or the prion, which are the good one, which don't cause disease, you have to know and to follow the way that proteins are made in the cells. And as I already said, the protein is a product of a gene. So the gene has a sequence which tells the cell the order, the name of each of the amino acid and their order in the molecule. So that gene is turned into an RNA form, which is called the messenger RNA. And the messenger RNA is read in a way, is going to turn the code into protein thanks to a micro machine which is called the ribosome. In a ribosome, which is inside the cells, the mRNA is threaded through the ribosome, in fact, and each time one of the code for a given amino acid goes inside the ribosome, the amino acid is cut from the cytoplasm of the cell and add to the protein which is being formed. So you end up coming out of the ribosome, you end up with a chain of amino acid, which I alluded to before, a linear chain, which is a sequence of different amino acids which correspond to the sequence that was in the messenger RNA, which went through the ribosome. So the ribosome is really a machine which turns the genetic code into the protein itself structure. And once this chain has been formed by the ribosome, chain of amino acid, as we already talked about a couple of times, it has to fold to acquire a structure and to acquire function. A chain floating like this is nothing for the cell. It doesn't have any function. It has to fold and make a three-dimensional structure which has a very specific shape and has some chemical property which will make the protein able to perform its various functions. So that's the background about how proteins are made, which is very basic molecular biology that you would expect people to have heard already, but I try to summarize that in the book to make sure that everybody is on the same page.
Yeah, no, you summarize it extremely well, and I think it's obviously super important. I guess moving a little bit more larger scale, so for some of the neuroanatomy, well, obviously you don't have to kind of walk us through all of the brain, cortical, subcortical areas, but in terms of understanding proteins and prions disease, obviously you're probably going to want to talk about the basal ganglia, which is the kind of cluster there in the subcortical region of the brain. What are the parts of the brain or areas that are predominantly implicated for some of the diseases like Parkinson's, like Alzheimer's, like Lewy bodies, and how prions work in those parts of the brain? Maybe you can also talk about, action potentials if you want. It's a little bit more specific, or the role of inflammation on some of these disorders in the brain.
Okay, the prion, depending the disease, will affect specifically a different group of neurons. In the case of the spongiform encephalopathy, of Creutzfeldt-Jakob, etc., it's a little bit all over the place. It's the neurons which are affected. So the neurons are, as we know, the cells which are performing the major job for the brain by transmitting information from cell to cell and computing this information. So the neuron degenerate, die, and the protein, which is also something we haven't started talking about, but which is very characteristic of all those prions, the protein aggregates. It makes some masses of protein outside the neuron, actually, in the case of PRP, but the neuron eventually die. A little bit all over the place in the cortex, and etc. Now, for some other diseases, the area which are affected, especially at the beginning, are more specific. In the case of Parkinson, for example, it's neurons in a small area called substantia nigra, which degenerate and which contain the prion protein. Although with time, it's going to diffuse away from this area along the axon of the neurons, and it's going to invade the rest of the brain, so that the first symptoms of Parkinson's disease are usually movement disorders, because that substantia nigra is controlling the movement of the limbs, the legs, the arms, etc. But later on, the prion is going to diffuse, traveling down the axons, diffuse to more part of the brain, and eventually cause different kind of symptoms, including dementia. I mean, unfortunately, fairly large or not negligible proportion of Parkinson patients will end up having some cognitive problems, including dementia. Yeah, so I was going to say, in Alzheimer, it's very specific area at the very beginning, but different from those of Parkinson. The lesion starts in a small part of the brain, which is called the entorhinal cortex, which is involved in memory, especially in acquisition of memory, of short-term memory. And from there, again, it's going to diffuse to more part of the brain and cause the dementia that's characteristic of Alzheimer. So this raises some really interesting questions, which are being studied very actively in several laboratories right now, which is why prion for Parkinson, which is alpha-synuclein, starts in substantia nigra, because alpha-synuclein is present all over the brain, not just in those neurons. And why prion for Alzheimer, A-beta 42 and tau, starts in entorhinal cortex, because A-beta, the protein that is a precursor for A-beta, and tau are present all over the brain again. So there are some specific aspects of the neurons in this area versus that area, which makes them susceptible to having those proteins turn into prion and starting the disease process. So if we could understand what is different about the neuron in this place versus that place, which makes them susceptible to this prion or to that prion, we can learn a lot about the mechanism of the beginning of the disease, of the mechanism which triggers the disease, and hopefully can interrupt that mechanism to interfere with it. So there is a lot of activity being done in understanding what are the molecular difference between neurons in this place or that place, which makes them susceptible to this type of prion or another type of prion. And this is, we talked about only 3 different diseases, but there are more. So each one has its own specific way of favoring one of their protein to be turned into a prion, which is a very, very important and fascinating question.
Let me ask about these differences here. Again, sort of a kind of way in which it's organized or structured. So you've talked about the prions in Parkinson's. I was trying to understand and navigate some of the differences. There starts to become this overlap here. But we have Lewy bodies, we have Parkinson's, we have prions disease, we have multiple system atrophy. And prions are implicated or can be in some sense or adjacent to it in all of these. How do we have Lewy bodies as a disorder, but then also Lewy bodies implicated in Parkinson's? Like, what's the kind of overlap here? But how do we see these things as different? Sometimes people will say prions disease as different from, Parkinson's or multiple system atrophy. How do we understand what we're discussing, especially in a medical context or even in a research context, if there's a lot of overlap here with these things?
Yeah, so Lewy body is just the name of a lesion that you observe under the microscope, which was described by Lewy, a German pathologist, more than a century ago, I believe now. So Lewy body is not a disease. It's just this particular lesion that you observe in the brain of patients. Now, Lewy bodies, we know now, well, we have known for some years now, but it was an important discovery that people were able to purify those Lewy body out of brain of diseased patients and see what they are made of. And they figured out that the main component of a Lewy body is a protein called alpha-synuclein, which turned out to be a prion protein. So Lewy body is only the definition, the name of those lesions, which are made of aggregated, misfolded alpha-synuclein and some other protein, but the main protein is alpha-synuclein in Lewy body. Now, there is the confusion can come from the fact that there is a disease called Lewy body dementia, and this is again due to alpha-synuclein, which misfold, aggregates, and form Lewy body. But those Lewy body, instead of in Parkinson, they are, as we said before, they are essentially in substantia nigra, the neuron of substantia nigra. In Lewy body dementia, they are in different places. They are in the cortex of the patient. Now, I already said that, unfortunately, a Parkinson patient quite often end up having some cognitive problems, including, unfortunately, some dementia. So, Lewy body dementia is, in a way, a form of Parkinson which starts with a dementia, but the protein involved, alpha-synuclein, is the same in both, and it, in both cases, aggregates inside the neurons and makes those inclusion, those structures, which are called Lewy bodies. Now, you mentioned also multiple system atrophy, MSA. So that's an interesting disease, which is also called by alpha-synuclein, the same prion, but instead of this prion, of this protein misfolding into a prion in neurons of substantia nigra or neurons of the cortex, in Parkinson or in dementia. In this case, this protein misfolds in a totally different cell called an oligodendrocyte, which is a cell which makes a myelin, which is a sheath that surrounds axons. Now, the disease, therefore, is totally different because it's not neurons which are dying and which are affected, it's oligodendrocyte. So you have a totally different disease because the protein misfolds in a different type of cell. But it is the same protein. So depending where the prion is going to occur, the misfolding is going to occur, and the prion is going to accumulate, you can have totally different disease if one case is a neuron and the other case is a different cell, which in this MSA, multiple system atrophy, is oligodendrocyte. Does that clarify the issue there for you?
Yes, it's very, very good. It was very, very clear because these things start to kind of overlap. I've learned a little bit in my own training, and so I was like, but it's still kind of a sort of a labyrinth to coordinate through. But it does sound like the placement of where these proteins are happening, or misfolding, if you will, and in the case of multiple system atrophy, in different another kind of, not in a neuron, but it sounds like you're saying in the myelin, which we see in things like multiple sclerosis, which has issues with myelin. So it's very interesting how the protein is in everything, or can be in a lot of these things, these diseases, but it just is really seems like on the placement of it is really, really particular. And then also how it will manifest, I guess, in the various symptoms or symptomatology as well, which is also kind of dynamic. Let's go back to Alzheimer's. You talked about Alzheimer's a little bit and how prions are implicated there. You can mention the involvement of that. Maybe some people will know about ACH, acetylcholine. You can talk about the APOE4 gene, amyloid plaques, APP, neurofibrillary tangles. These are kind of specific to Alzheimer's that we know, or not just specific to Alzheimer's, but they're implicated there. How is the tau protein involved with Alzheimer's, and how does that work?
Yeah, that's a complicated field. Again, it's a neuron problem. Again, protein misfold. Protein which are everywhere. I mean, tau, for example, is a protein which plays an essential role in Alzheimer. It is present all over our body. Many, almost every cell in our body uses tau for one reason or another. But it turns out that in some patients, this protein is going to misfold in neurons, although it's present in different kind of protein, in different kind of cells. But in some neurons, in some specific neurons somewhere in the brain, this protein tends to misfold and cause Alzheimer. So in Alzheimer, two proteins misfold and turn into prion. Tau, which I already talked about, which is inside neuron, which is important for all sorts of things, especially the transport of protein down axons of the neuron. And A-beta 42, or amyloid, people have heard that, different names for that. This is also a protein which is present in many neurons and other cells. Now, that protein is at the surface of the cell. It's not inside, it's at the surface. And in Alzheimer, this protein is cleaved, is cut from the surface, and therefore now is floating around outside the cell. And there it can misfold, so it's only a portion of the protein which is cleaved off. And there it can misfold and start behaving as a prion, which means, and we haven't said that, I think, since the very beginning, and it's essential. What makes a prion protein a prion is that once it's misfolded, it's going to misfold in its own shape more copy of the same protein. So the thing accumulates, and that's how you end up having an aggregate of protein. So to go back to this amyloid protein, it's at the surface of the neuron, it gets cleaved, so you get a small piece which floats now, and which, by behaving as being a prion, starts to misfold other pieces which have been cleaved from the same neurons and accumulate outside the neuron in a big aggregate, which is called amyloid plaque, and which shows again in the microscope when you look at the brain of deceased patients. So you've got two prions here in Alzheimer, one which accumulates and behaves as a prion outside the neuron, and one which is a beta, and one tau, which behaves as a prion but inside the neurons. So it's a complicated disease. And which is really responsible for killing the neuron eventually and therefore causing the disease? Is it tau? Is it a beta? This has been a story that's been going on for years. It's now very well accepted, very clearly shown that the amount of disease, which is really the severity of the dementia, goes along, parallels the amount of tau which is present in the neuron of the patient. So the more tau you have, the more dementia you have. So tau seems to be clearly involved, related to the extent of the disease. But without a beta, you don't have the tau folding into a prion. So a beta seems to be what starts the tau protein to turn into a prion. So you accumulate a beta, and many people who don't have Alzheimer have accumulated a beta in their brain, but without having any dementia. But in some cases, this A beta is going to go back into the neuron, be internalized by the neurons, and there it is going to favor the misfolding of tau. And now you start something which is going to cause a disease, and the extent of misfolding of tau is going to parallel with the extent of the clinical disease. So the two of them are important, but they are important for the disease, but they are important in different ways. Now, if you want to prevent or treat the disease, you may want to act on both, on A beta, which seems to be very important for triggering the misfolding of tau, or on both if possible. And that's why now there are several clinical trials ongoing and drugs on the market, actually, at least in the US, to try to eliminate the A beta, the misfolded A beta in the brain, and to try to eliminate or prevent the accumulation of the misfolded tau inside the neuron. Now, those clinical trials or those drugs are just at the beginning of the story, and it's very clear that it's a very important avenue for treatment. But it's also very clear that we are only at the very beginning of finding the right drugs to do that. Right now, the drugs that have been tried are essentially antibodies which bind to those misfolded A beta or tau, and which favor their elimination by the body. But there are other ways to interact, not just antibodies. You can engineer drugs which will specifically bind to those misfolded protein and prevent them from behaving as prions. So that's also a very promising avenue, which is not yet in clinical trial, but which may be actually the way for the future.
Yeah, that's a very complex story there on the inside and the outside, and where the future is going. Obviously, there's lots of work there because it's an awful. All of these are kind of awful diseases. I'm curious. I want to ask you about yeah. Well, that's the other problem is how often we're seeing this so often. We gotta figure it out, of course. But let me just ask real quick here about where do we see, as you said, proteins are everywhere. And where do we see, I guess, prions being implicated in other things such as Lou Gehrig's disease, Huntington's disease? Do we see it in MS, multiple sclerosis as well? How does it look? Because a lot of it is what you're saying is we're seeing these proteins and they're located in different parts, substantia nigra, or it's in the neocortex, or it's here, and it folds or misfolds differently, and so it presents differently. How do we see prions in other types of diseases such as Huntington's or maybe even multiple sclerosis?
Multiple sclerosis, there is no prion that has been described. So far, I mean, maybe somebody will find one, but we don't know of any prion protein involved in MS, in multiple sclerosis. But the other one that you mentioned, yes, ALS, which is myotrophic lateral sclerosis, people know it sometimes under Lou Gehrig disease. It's different. There are probably at least two proteins, a bit like Alzheimer, but different protein, which also are involved as prion protein in ALS. One is called TDP-42, the other one is called SOD1. So we know less, but there is very active work being done about the mechanism of the disease and what the prion protein does to kill the neurons in ALS, which is also a typical example of those things. Those proteins are everywhere, but they only turn into prion and cause a disease in very specific group of neurons, sorry. And in ALS, as I'm sure know, it's the motor neurons which are affected, so people get paralyzed. So why in the motor neurons and not in other kind of neurons? Why does TDP-43, which is all over the place, turns into a prion only in those motor neurons? Again, same problem that was addressing before with in Alzheimer. Why in the neurons of the entorhinal cortex does tau turns into a prion? So same question, which will have different answers, obviously, but very important to try to figure it out. So that's for ALS. You also mentioned Huntington. Huntington is an interesting disease. It's a disease of the neuron again. It's a different protein, a different prion, which is called huntingtin. And it's interesting because it's also a genetic disease. A genetic disease, it's caused by mutation in that huntingtin protein, which gets elongated. The genes get elongated, and the protein therefore gets elongated, having more amino acid than a normal protein. And after a certain number of, a certain size of this elongation, this turns the protein into a prion. It misfolds and turns into a prion. So again, why does it happen in some neurons? We don't really know. The mechanism is not really understood. Although there is a lot of work and there are a lot of results too, but I'm not sure that we can really make a clear picture right now about the mechanism of the disease in the case of Huntington. And then there are some diseases outside the brain also, including diabetes type 2, which is a very common disease where a protein in the pancreas turns into a prion. So it can happen in many different diseases and in many different organs. And we probably don't know yet about others, which could be in other organs that we haven't really investigated.
Yeah, that's I didn't know that about with diabetes. That's interesting, and I can as these things are more common. Alzheimer's is common, unfortunately. Diabetes is common. Obviously, behooves folks to try and figure out how we can prevent these things. I'm curious about in terms of you kind of give it towards the end of the book. This kind of memory overview, how memory works. Obviously, there's working memory, prospective memory, long-term memory, short-term memory, and then respective declarative implicit, explicit memories, things like that. But instead of that, maybe tell us. You mentioned the Candle's work with aplasia, the sea slugs, for
Yeah.
long-term potentiation, protein synthesis. I guess help us understand here how you can say what it is, CPEB, what that is, but how it's a good protein, and how there are other good proteins, or excuse me, not proteins, prions. How it's a good prion, and what are some other good prions? Again, using the good kind of loosely, but here we've been talking about these kind of
Right.
malformed or oddly shaped or misformed kind of folded prions. But how does the CPEB seem like a kind of maybe more positive or helpful kind of prion?
Yeah. So a prion, the one that causes disease, as we already said several times, is a result of misfolding, which is that the protein, to have its normal function, has a certain shape, and because the folding process of a protein is a very complex, very, very complex situation. You have a long chain of amino acids. They are different. They have a given sequence, and they fold in a stable form. Now, to do that, the different amino acids interact with each other, and some of these interactions are strong and stable, and some are weak and they won't stay for very long. And eventually, the strong one will form the shape of the protein. Now, this folding process, which is very complex, which right now we can predict with some very powerful computer programs, but this is a major advance in protein biology. For a long time, we couldn't do that. We had to solve their structure by complicated techniques. We couldn't predict it from the sequence. So this folding can, again, misfold and create a prion protein, which is a protein which is going to have a shape that will give the same shape to other copy of the same protein. And therefore you accumulate the misfolded protein and it becomes toxic. Now that's the misfolding. But the good prions, which were discovered a little bit by accident in a way, like many things in science, in fact, it's the same protein, but now we don't call it misfolding. It's a protein which are different way of folding. It can fold in a given way, which is going to be what we talked about, and have a function under this particular shape. Or it can fold in a different shape. And that shape is a prion, which means that it's going to, again, accumulate, cause the folding of other copy of the same protein into its own image, its own shape. And this is going to be used by the cell because now the cell has two different functions with the same protein. So it's a way of having more power with fewer genes, because a single gene which make a given protein can then give you two different folding, each one making a different things, having a different function. And one of the folding is going to be a prion, which will accumulate, and that's this accumulation of that form is how it's going to perform its function. So all of that sounds really, without making pictures, without seeing it, it sounds very abstract and complicated. I'm sorry about that, but that's what it is. And we now start to realize that those proteins which fold into a prion to perform a function, not to cause a disease, they are probably everywhere. They have been around with life for a long, long time. You find them in yeast, in primitive organisms, even viruses can use that. So it's been with life forever. And because it's been selected by evolution, because it performs some functions, which, for the one that we understand so far, has essentially to do with adjusting the functioning of a cell, of an organism, to a change in environment. For example, a bacteria or a fungus, yeast, which will find in its environment a new food, a new sugar, or new something, needs new enzyme to perform, to use that new source of energy in its environment. And one way, it's not the only one, but one way that evolution has figured out to make this change in function is by turning the protein into a prion and changing its function. Okay, so those are not misfolding, they are good folding to perform a function. And they are not due to chance, or malchance, like the one that cause disease, which happen with time because you're getting old, and then the probability of misfolding increase with time. No, they are induced, they are caused by a change, for example, in food in the environment for some yeast, or something else happening, which specifically turns the protein into a prion. And this is what was discovered in two fields. One was in parallel, more or less. One was the yeast, a geneticist, and they described a phenomenon that I couldn't explain by classical genetics, and which is due to a protein turning into a prion. And the other one is the neuroscientist, and you were talking about Eric Kandel, a very important and brilliant neuroscientist studying memory. Now, memory is, of course, one of the very important functions of our brain. As you mentioned, there are different, we divide memory in order to be able to study it in different sort of classes of memory, including short-term memory, long-term memory, etc., etc. Short-term memory, especially the acquisition of short-term memory, is unfortunately what disappears first with Alzheimer, so that we don't remember what we have just been doing, and we are just unable to deal with the everyday task if we don't have short-term memory. Memory, of course, poses very complicated and important question. How do you acquire memory? What is happening in the brain when you acquire memory, when you store it, and when you retrieve memory? And this is, of course, retrieve memory is in the case of long-term memory. Memory of things which happened many years ago in your childhood and which you've totally forgotten about, but it can be triggered to be reactivated, and it's still there, it's come back. So in what kind of form does it stay in the brain for so long, and how do you acquire this form that keeps the memory in the brain? This has been a long time debate among neuroscientists, philosophers, etc. And we are now making tremendous progress to understand that. It's a very spectacular aspect of neuroscience, fascinating. We know that, and I'm going to talk about essentially long-term memory here. We know that memory consists in building a network of neurons which communicate with each other from very different parts of the brain, and which is more or less stable and is going to remain like that for some time, or sometime very long time. Each neuron is connected to another neuron by what's called synapses, which are contact between the two neurons. And those contacts are where the information, the stimulus from one neuron, is triggering a new stimulus in the downstream neuron. So there are the places where one neuron excites another neuron, synapses. Now, any neuron, and that varies very much depending which kind of neuron and which type of animal or human you're talking about, but each neuron has many synapses, thousands of synapses. In our brain, the number of of synapses in the human brain is absolutely astronomical. There are more synapses in our brain than there are stars in the Milky Way, in our sky here. So it's in the billions, of course. So each neuron has thousands of synapses, and only one or a few maybe will be involved in a given memory. So the same neuron can be involved in many different tasks and many different memory, but it will have one synapse which will remain stable, and this synapse is going to be part of a network which will constitute the physical substratum for a given memory. This network is sometimes called an engram. Now, what Eric Kandel found using a very simple animal, which is a sea slug, which has few neurons and very large ones, so they're easiest to study, is that acquiring memory doesn't mean making new synapses. It means using the synapses which are already there, but making them stronger. So acquiring memory does not create a network that didn't exist before. It reinforces connections which were already existing and which now are going to be stable for a long time. So that was a very important discovery. So the problem becomes complicated because then you have a neuron which has thousands, 100,000 sometimes, different synapses. And for a given memory, it's only one of those synapses which is going to be made strong and stable. So how does the neuron knows which synapse has to be strengthened when it is in the process of acquiring a memory? That's been the major problem for a long time in the field of neuroscience and memory. And what Kandel discovered, and I'm going through many, many different steps there to go to the end of the story, is that what happened is that to make a synapse stronger, you need to add a lot of different protein to that synapse, which is going to make it bigger. You can see it bigger under the electron microscope, which is going to be changing the structure of the synapse and make it more stable. Now, you need protein to be synthesized for that. And the neuron has to bring those protein or make those protein only in one synapse, and not on the other one, which would, of course, scramble everything. So how does that happen? How does a neuron know this synapse has to be reinforced and not its neighbor or whatever? What Kandel discovered is that during acquisition of memory, the synapse is being used. And if you stimulate the synapse repeatedly during acquisition of memory, one of the protein which is already present in the synapse, which is called CPEB, turns into a prion. That protein turned into a prion is going to recruit and turn into prion, as I said before, other copy of the same protein. And that's going to cause an aggregate of the CPEB protein in its prion form, which is big and which is going to be maintained, because even if it's degraded a little bit, it's still going to be able to fold again into a prion form other copy of the protein. So it's going to be stable in time. And this is a tag. This is what tells that neuron that this protein now has been stimulated repeatedly and under certain conditions during learning, and it has to be enlarged and it has to be made stable. And it turns out actually that the prion form of that protein favors protein synthesis, favors making more protein there. So all of that was discovered with this very simple sea slug aplysia, because it was easy to study in the laboratory. But it also is present in all sorts of organisms. It has been extensively studied in the fruit fly because it's a very useful system for geneticists and molecular biologists. It's been found in mouse, etc. So it's most likely at this point that it is a general phenomenon which is present in all form of life which can acquire a long-term memory. It's a very fundamental discovery of a good prion. And a fascinating, elegant scientific achievement.
Yeah, I mean, it's obviously, I mean, I really, really appreciate you explaining all that, because it's very important to understand how there are many things historically and more presently of understanding where, things land and where they reside, and trying to understand how this is helping us understand things for the future, which leads to kind of my final question here is, what do you see with the future of medical research, but also where things are at now, right? So where things are at in medicine, maybe on the research side, I guess. Where do you see things kind of going in the next 5, 10, 15, 20 years on trying to do important work, whether it's with different treatments or just understanding etiology or other things with prions, and how we can try and have a better handle on them, maybe on the knowledge-based side, but on the treatment side as well?
Yeah, it's of course, very important question. I'm glad you asked it. I am an optimist. I see things going very well. I mean, if you think about what's been achieved in the past, not even century. Actually, I have some notes about that. I'm thinking about writing about it. In 1944, Schrödinger, a famous physicist, discovering elementary particles, etc., turned his attention to life. And he wrote a little book that many people have read, I'm sure, and which is very well known, which is called What is Life? 1944. Now, at that time, we didn't know about DNA. We didn't know about genes in terms of what are they made of. We knew genes because of Mendel's story, etc. But in terms of molecules involved in life, nothing. We knew nothing. Watson Crick and the structure of DNA in 1953, 10 years later. And see where we are now. And if you see not only where we are now, what we have accumulated since the 50s, but the rate at which we have accumulated it can be plotted, and there are many of those plots that you can find in the literature about discoveries according to the years going along. Well, the rate of fundamental discovery, I'm talking about biology only, of course, is, if not exponential, it's almost exponential. I mean, it's increasing at an incredibly fast rate. And neuroscience is certainly one of those places where it has been going at an incredible pace. Now, that's not even 100 years. Now, when did really scientific inquiry start with humans? About, 3,000 years ago, 4,000 years ago? And how many years are going to be human on the planet? I hope many, many, many thousand years. So what's going to happen? It's clear that we are just at the very beginning. We're going to understand things in much, much more fine, precise, and detailed molecularly and in all sorts of aspects, ways than we do now. And so if you turn towards medicine and what will happen is that all medicine is going to be in great part based on our understanding of the mechanism of life. finding drugs, developing drugs, needs to understand the mechanism and finding a target and understanding how the molecule behaves so that you can design something. And we are at the beginning of designing new proteins. Only started 2 or 3 years ago. Already won a Nobel Prize this year for that. Designing new proteins which make new functions that didn't exist before. So we are only at the very, very, very beginning. And at the rate at which things are going, I think there is all reasons to be extremely optimistic that scientific progress in biology and in other fields as well, but in biology, and also the fact that things are getting integrated more and more between fields which were not really talking to each other before. If you think about neuroscience, since most of what we've talked about has to do with the brain, neuroscience, a different aspect, of course. It goes from philosophy to molecules. Until not very long ago, they were in the center of it, there were some cellular and molecular neuroscientists, the people studying neurons, their function, how they work, how the influx is transported, transmitted, etc., the regulation. All right. On one side of that, without much interaction, was psychology, even sociology. I mean, people looking at the brain, not in terms of molecule, but in terms of what do they do, even if we don't understand how they do it. How do they look at things? On the other side of that was people looking at the brain as a computer scientists, saying, well, neural network. Okay, we're building neural network in a computer, and maybe if we understand what that happens in a computer, we can translate it to what's happening in the brain. So there were really those 3 different aspects, and the scope of things is enormous. I mean, people in psychology talking to people in artificial intelligence, it's not very easy. But those 3 fields are now really fusing very actively, and very very spectacularly, very with a lot of potential. So I think I am terribly optimistic. I think that things are going to move very, very fast, and medicine is going to profit from that immensely.
Yeah, well, we can only hope, right? We can only hope that we're going to, accelerate some of these things and be able to find good treatments. And I'm always kind of marveled at what good research and science can do. So, Michel, this has been a absolutely wonderful conversation. This has been so informative. It's been so helpful. Really, really appreciate you coming on the podcast and talking all about it with us. So big thanks.
Thank you very much. I appreciate it. I liked it.